Hi, well, thanks for joining me today. I'm Kerri Phillips, and I'm a clinical product manager here at DNASTAR. And today we're going to talk about our next-gen sequencing software for cancer gene panel analysis. So today I'm going to talk a little bit about DNASTAR history and our software, and then I'll get into working with next-gen sequencing data and the importance of accuracy. And then I'll get into a detailed workflow for cancer gene panel analysis. So this year at DNASTAR, we're celebrating our 30th anniversary. DNASTAR has been making software for life scientists since 1984. In this infographic here, you can see on the lower left-hand side an image of our founder and president, Dr. Fred Blattner, who launched DNASTAR from the work that he was doing in his E. coli genetics lab at the University of Wisconsin. Now, back in 1984, it was quite an accomplishment to be able to run powerful genetic software on a desktop computer. But for 30 years, that's what we've been doing, and we've been continuing the tradition of providing powerful software on accessible and affordable hardware. And then last year, we took another leap forward and made all of DNASTAR software available also on the DNASTAR cloud. And so this makes the suite of products available to life scientists regardless of what hardware they might have in their lab, which is helpful for some people with next-gen analysis, which requires a little bit more computing power. DNASTAR is consistently cited more often than other software in peer-reviewed articles. We've had a tradition of this, and even up to last year, you can see that DNASTAR software is cited twice as often as even our nearest competitor in these publications. Another advantage of DNASTAR software, our next-gen suite of products can handle any size project and any of today's next-gen sequencing technologies. So with our software, you have the flexibility to run projects from targeted regions and exomes right on through multiple genomes or metagenomic analysis. And you can use your Illumina, Ion Torrent, or any of today's next-gen sequencing technologies. Another advantage of DNASTAR software is the wealth of resources that are available to our customers. So right on our website, you'll see that we have 3 years of pre-recorded webinars on there on a variety of topics. We also have a complete library of software tutorials, and these are sortable by, you can search for them by product name or by topic. And then we have over 200 videos in our video library online. And these videos are all very short and sweet. They're less than 5 minutes long. So if you're new to DNASTAR software or you're doing a workflow that you're not used to, these 5-minute videos are designed to get you up and running, give you a jump start on your workflow and get you started. And then if you ever feel that you need to talk to a live person for that one-on-one consultation, we do have live chat on our website, and you can find the chat live button throughout the website and get a hold of a real person on the other end. This DNASTAR software is organized into suites around different types of research. So for example, we have the evolution suite. We also have our structural biology suite, which gives you some 3D protein analysis as well as 3D protein structure prediction. And in that suite is included the NovaFold for structure prediction. And today I'm going to be talking about our 3 core applications in the laser gene genomics suite. And in here we have all the applications that you would need for your next-gen sequencing assembly and analysis, all integrated in one useful suite of products. So let's get into some of what you'll need and what you'll find when working with next-gen sequencing data and gene panels. with gene panel data sets, you might be working with kits from, say, Torrent or Illumina. With the Torrent, for example, the AmpliSeq Cancer Hotspot or Comprehensive Panel, you'll get some files typically. These might include an unaligned BAM file, bed files, and VCF files. And I'll talk a little bit more in detail about these types of files in just a second. With Illumina, you'll get a FASTQ file. Often these are paired. Bed files, Illumina will sometimes call these manifest files, and VCF files. And then if you're working with a custom gene panel, you might not have these types of files. You might be maintaining your targeted regions or your annotations in a spreadsheet. And I'll talk a little bit about how you can bring those in as well. So what are VCF files? Well, VCF stands for Variant Call Format. And these you can get from a variety of sources. Like I mentioned, they might come with your gene panel. You might also find them in 1000 Genomes, Exome Variant Server, or other software. For using them in DNASTAR's LaserGene SeqMan engine, you can import a VCF file to supplement the dbSNP information that might be in your genome template package and otherwise annotate. You might also have your SNPs in an Excel format and not in VCF. If this is true and you'd like to import them, DNASTAR does have a tutorial that's available, and this can walk you through converting to a VCF file format. Both LaserGene SeqMan Pro and the ArrayStar applications can save their SNP table information in VCF format for export into other applications. Bed or manifest files, these are tab-delimited text files, and these contain a list of the targeted regions that you're looking for in your gene panel. Again, the file formats can differ depending on where you get them. SeqMan Pro will use a bed file to filter your SNPs or your variants outside your targeted region. So the bed file is really helpful to help you in the software determine how your coverage of your targeted region went, help you refine your coverage if you're still working on refining your gene panel. And if you have a custom panel, again, DNASTAR does have a tutorial available to help you convert, say, an Excel spreadsheet into a bed file. So accuracy is really important with next-gen data and especially with gene panels. So how can you be sure that you're catching the variants that you're looking for? Well, one way to do this is to use a well-characterized reference sample. Sometimes we call this a gold standard, along with a list of your known variants within that sample. So one example of this is coming out of the Genome in a Bottle consortium from NIST. And what they have provided is this sample from that they label CEU NA 12878. And this is a female person from Central European descent, and they have taken her genome and sequenced it using a variety of sequencing technologies, I think half a dozen different technologies. Then they've come up with a consensus of which variants are in there, and they have a high confidence of over 80% of that genome. So it's a very useful standard to use to run a validation test. And for example, in the DNASTAR software, we do have a validation control workflow for gene panels. And this can be used to calculate accuracy. So how are those calculations done? Well, you start with your known positive variants, and you see how well your analysis went and your variant calling, and you get a true positives number. And down here I have some examples of an actual validation run that we did using Illumina data and a sample of the NA12878 DNA. So then we have the false positives. You can then calculate a false positive ratio a false discovery rate. And from those numbers, then you can calculate your sensitivity, which is a measure of the true positives, and then the specificity, which takes into account the true negatives. And from those two, then you can have the balanced accuracy. And you can see that we had very good luck with that. We had over 99.8% accuracy with our Illumina data. So that's just one example of how you can use a validation control to ensure the accuracy of the gene panels. Some other tools that DNASTAR software offers can help to improve accuracy. So in combination with the validation report, you can also use DNASTAR tools to see how specific your primers were in your gene panel design. You can see the efficiency of your sequencing instrument and your sequencing technology. You can see the accuracy of your assembly, of course, and your variant calling. And then you can also use it to evaluate the effect of different SNP filters that are applied after the assembly. So here's a flow chart of how you might use the validation control workflow in DNASTAR software. So here at the top and to the left, you can see that we have used a bed file of the high confidence regions for that NA12878 sample, and we've crossed it with the targeted regions for the gene panel. Then likewise, we took the bed file and we crossed that with the VCF file that had all of the known variants for that sample. And with that, we got a VCF file that was specific to this particular gene panel. And then if you go in the bottom half, you can see that SeqMan Engine can take the sequence data, the NA12878 FASTQ file, and can be fed into the SeqMan Engine. The variants are called and the references made based on the VCF file. And then from that, the validation report is automatically generated. So that's just one way that you can use the validation control as part of the gene panel workflow. And there you can see that we did get very high accuracy using that, and we're able to measure that over time and get continuous accuracy metrics with every run then using that same procedure. If you'd like more information on that, we do have a paper, SeqMan Engine as a high accuracy NGS assembler, that is available for download on our website. So let's get into the details of the cancer gene panel workflow in LaserGene. Here's a really simplified flow chart showing what I'm going to walk through today. So what's nice about this workflow is that it allows you to load your normal control data and your tumor data in one step as part of the workflow. And then the engine software will calculate the normal variants as well as the tumor tissue variants. And then with just 3 easy filtering steps in the ArrayStar application, we'll get our tumor-specific variants. So in DNASTAR, we have our DNASTAR Navigator, which is sort of our hub for utilizing all DNASTAR software. And you can see here that they're organized again by the type of research that you're doing. Our next-gen sequencing applications are grouped together. And today I'm going to walk through SeqMan Engine, SeqMan Pro, and ArrayStar. Another nice thing about the Navigator is it's also a central location for getting more information for each of your applications. So, for example, if you hover over one of the applications, you'll get a pop-up window with active links to more information, such as our videos on the website, tutorials, or practice data. So the first step in our cancer gene panel workflow is to launch SeqMan Engine. So here's SeqMan Engine. SeqMan Engine is wizard-driven, and it walks you through all the steps that you need for your assembly setup. So the first step is to select the radio button for Create a New Assembly Project and click Next. And then the next screen shows you all the different project types that you could run in SeqMan Engine. We have everything from metagenomics through genome assembly. We do also have 2 gene panel workflows. One is a Mendelian or germline gene panel assembly, and then today the cancer somatic gene panel assembly that I'm going to show you. So I select that radio button and click Next. And for assembly type, then we have a choice between a normal workflow or a templated assembly with control. And again, our normal patient sample will be our control in this case. For our project file setup, we need to give the project a name and select a folder for the output data. For our genome templates and bed files, we use the input template file screen. Now, DNASTAR on our website, we have curated several genome templates for model organisms. So we have a wide variety. We have dog. For all of them, then we've incorporated the dbSNP information, so that is loaded automatically when we use one of our templates. For the human, you also get the cosmic and GERP information automatically. So going back to the screen, the genome template and bed file screen, I've selected the genome template in the top middle window there. And then down here you can see that I have selected targeted regions file, and I've uploaded a bed file. And then click Next. For our input sequence files, then we can select what sort of read technology we're using. Now I've got some paired reads of Illumina technology, but you can see that in SeqMan Engine I could load 4x4 or Ion Torrent or PacBio as well. So down here I'm going to give these paired reads names so that we know that we have the tumor data together. So I give each read a matching name, and we have some tumor sample data from patient number L004, and then we have 2 set of data that actually came from blood from that same patient. And then to set up the experiments, this is where we tell the software what is our baseline or control. So you can see here that in the control column, I want to select that the normal sample is the control, and then I want to tell that it's a baseline type of control. And then we also want to assign a VCF file here. So here's the VCF file with the SNP annotations for this gene panel. So now we're getting to our assembly options screen of the wizard, and we're almost done. As I've been going through, I've been making selections, and based on those selections then, SeqMan Engine will optimize several settings, and this is based on the wealth of knowledge that goes into knowing what works best for different types of data and different situations. So typically there's no need to make any adjustments here. However, if you did find that you wanted to make some adjustments based on the type of work that you were doing, you certainly can do that. Another option is to select the advanced assembly options, in which case then you get this additional dialog panel with 3 tabs, and you could make some changes. For example, you might want to, by default, filter out SNPs below a certain percentage in your samples. And with that, then you're ready to assemble. So a script is created here in SeqMan Engine. You can save that out, or you can just click Assemble, which is what I did. And then about 45 minutes later, your assembly will be complete. Depends on the data size. This particular data set took me about 45 minutes for each, for the tumor and the normal. And I'll talk a little bit about how you can actually go even faster with that. So here is the completed assembly, and I have a project report. Then down here at the bottom of the SeqMan Engine Wizard are two options. I have the option to launch it in SeqMan Pro, or I can launch it in ArrayStar. And there are different things that you can do in each of those applications, and they're integrated right here. So the first thing that I'm going to do is click on Launch in SeqMan Pro. So here's SeqMan Pro, and really the strength of SeqMan Pro is to identify variants, analyze your depth of coverage, annotate your assembly. So right here I've opened the Tumor assembly project. So this is just one of the two that we just ran. And I'm taking a look here. This is the first screen that I would open. This shows all of our contigs. And in this window here, you can see that we have contigs for each of the chromosomes in this sample. And if I highlight that, and then I can look at the SNP report for the entire assembly. And here's the SNP report. Wealth of information in the SNP report. It's a table with many, many columns of information. From here, I can see, for example, things like the SNP percent for each variant that's called. And over here, a new feature in LaserGene 12 is this ability to view SNPs that are next to each other in one coalesced view. And what I've done is I've clicked on the twisty triangle there on the left-hand side to expand this view, and then you can see all the coalesced SNPs underneath, but I don't have to scan through the whole list to see these that are side by side. So another very nice thing in the SNP report view is the ability to further filter and then in real time see the effect of these filters on the SNP report. So I would click on Filter button, and this brings up the SNP filter criteria. You can see I have a wide variety of criteria that I could use here. Again, I could, if I were to increase the minimum SNP percentage here, I would immediately back in the SNP statistics report see a reduction in the number of SNPs reported here. So that's always option. Very powerful way to quickly dig down and try to get to the variants that you're looking for. So then if I went back and I double-clicked on these coalesced SNPs, I would instantly be brought to the alignment view. And it would jump me directly to the place in the alignment that I was looking at. And so here you can see the deletion that's being indicated by that particular variant call. Another really powerful tool in SeqMan Pro is the strategy view. So this again is one of those areas for monitoring your quality. So you can take a look at your depth of coverage for your area that you're of interest. You can also look at your pair consistency. So very helpful as you're developing a gene panel or just to ensure that you're getting good quality results throughout the process. So then the other option when I had a completed assembly is I could launch in ArrayStar. Now the power of ArrayStar, one of its key applications is the ability to compare samples from data from multiple samples at a time. So for today's application, I'm going to be looking at 2 samples at once, but you could look at anything from 1 to 100 or more samples, and that's really what makes it so powerful. So for this project then, I'm opening both the normal and the tumor sample in one project. And the first thing, again, that I might take a look at would be the SNP table. And SNP table is really helpful because I can see, compare and contrast the information from both samples at once. So here you can see I have a column here for the normal sample, and then I also have a column side by side of my tumor sample. And right in the middle there, you can see, I'm sorry, I'm just going to get rid of that highlighting. And right here, then you can see where there's a variant that's in the tumor sample, but not in the normal sample. So I might want to take a closer look at that. I can hover over the classification column, get more information for that, and just see, well, okay, so at this point the depth of coverage was 66, and this particular variant is presenting at a SNP percentage of 13.63%. So just an interesting way to delve a little bit deeper into the data. So here's the powerful part of ArrayStar where we might run, start doing some filtering, comparing the two samples to try to get to what variants and what genes might be affected in the tumor sample that weren't in the normal blood sample. So the first step for this is to determine which variants we'd be interested in. So I'm going to take a look only at the tumor sample, and then I'm going to select Choose SNP Criteria. And that brings up a dialog box. This should look very similar. It's synchronized with the SeqMan Pro, very similar filtering capabilities here. And for this SNP set, I'm going to take a look only at nonsense and frameshift mutations. I'm going to make some adjustments here to the SNP percentage and to the minimum depth. I'm going to look at SNPs that are only at 25% or greater, and where the coverage is at 30% or 30 minimum depth of 30 as well. And then I'm going to click OK. You can see here that I get some variants that are called. I have 124 variants, and I'm going to save those for my next step. So I hover over the Remember Results as a SNP set, and I'm going to give this a name that's sort of long but helps me remember exactly what I did. So I'm going to call it Non-Synonymous SNPs in the tumor sample at a depth of 30 and a SNP percent of 25. So that's kind of long, but it helps me remember. And so now I'm going to go back to that same tumor sample, except for now I'm going to focus on the genes that are affected by those SNPs. And so in my SNP searching criteria, down at the bottom here, you can see that I'm using the SNP set that I just saved. So here are all those SNPs, and I'm going to be looking for only the genes in the tumor sample that are affected by the SNPs in this set. I click Next, and there we have it, 114 genes affected by those variants in the tumor sample. Well, so then the last step is simply to subtract out the normal affected genes from the tumor, and that should leave our genes that are indicated as likely suspects of being responsible for this tumor. So in the next filter, I'm adding to this, and I'm selecting the normal sample. And you can see here that these read the filtering capabilities in ArrayStar sort of read like a sentence, so it's very easy to understand what you're doing. So in the top row, I have find genes with SNPs in one of the tumor example, and then I have in the second row, find genes with SNPs of these criteria in 0 of the normal. So basically, if it's in the normal, filter it out. And then click Next. And sure enough, my genes have been reduced down to 11. So very quick and easy, 3 simple steps. I've now significantly reduced the number of genes that I would want to look more closely at for this. So to get a closer look, a little bit more detail, I could select the Select and Show Results in Gene Table option. And that brings up the gene table. So here again are the same 11 genes and in a table with a lot more information and annotations with regard to those genes. So again, here's in this data, you can see that this TP53 gene is indicated, which makes sense. It's a known gene, tumor suppressor gene. And if you hover over that, then you can get a lot more information about that. So just a really useful way to take a look at the information that you have and very quickly kind of winnow and sift down to what's important to you. So another tool in ArrayStar is the Venn Diagram. So again, just another way, nice image way to visualize the same subtraction. So on the right-hand side in the green sliver, we have, that's the normal genes, the genes that are found in the normal sample. And then on the left-hand side, we have the 11 same genes that were found only in the tumor sample. So that's pretty much the workflow for the cancer gene panel workflow in LaserGene data, or LaserGene application. So what are some other things to consider with next-gen data? Well, we have workflows in LaserGene. We have the specific cancer somatic workflow that I just went through. We also have the Mendelian and germline and the validation. But one of the first questions people ask is, how does LaserGene software perform? I mean, it's run on a regular, pretty normal desktop computer. So what sort of assembly performance can we expect? Well, here's some benchmarks. You can see that we could do a human genome in about 13 hours. We could run 8 human exomes in about 14 hours. TruSight Cancer Panel from Illumina with 10,000 X coverage, about an hour, or the lower X coverage AmpliSeq Cancer Panel from Torrent in about half an hour. So pretty good times. Well, how does this compare to other pipelines? Well, SeqMan Engine is a assembly and a variant caller, which is about equivalent to the, often compared to the BWA GATK pipeline. And so for this, we did some benchmarking side by side of the 2 pipelines. For the first one here, we used the Illumina exome. And then for the second one, we did an ion torrent exome from the HiQ. And you can see that typically the SeqMan engine had higher sensitivity in both cases. A little bit higher with the true positives here, a little bit lower with the false negatives. But I think what's really surprising is the very significant reduction in time that it takes to run. This is running the assembly and the variant caller in one step. So I think less than 1/4 of the time. With the Illumina goes down from, compared to 13 hours, the SeqMan engine can run that in 2 hours. And with the ion torrent data, we can get that down from 28 hours down to 6 hours. And what kind of hardware is needed? Well, it's pretty basic. I mean, we have a pretty basic, affordable quad-core computer with 16 gigabytes of RAM. Now, it does require both a scratch disk and some storage. And these can be internal or external storage or hard disks. but they do need to be separate hard disks. And we recommend somewhere between 1 and 4 terabyte scratch disk. I believe like 2 terabytes would be sufficient if you're running exomes. And then for storage, probably something over 1 terabyte of storage for next-gen data. But what if you don't have this type of equipment available? Well, you can also take advantage of on-demand computational power as well as data storage for next-gen data that can grow with your needs. So DNASTAR Cloud applications include the DNASTAR Cloud Data Drive, which can be used for long-term, secure, and encrypted storage. And then we have the DNASTAR Cloud Assemblies application, which is basically a SeqMan engine on the cloud. But one of the really nice things about the DNASTAR Cloud Assemblies is that you can run all your assemblies in parallel. So for example, when I was running the data for this example with the tumor and the normal, I ran both in parallel. So I got both done in 45 minutes using DNASTAR Cloud Assemblies. Whereas on a desktop computer, you would have to run them one after the other. So that sort of access to on-demand power can be really helpful. So here again are those benchmarks that I showed you, and you can see here that we can go from with the 8 exomes, instead of being 14 hours, you can actually get that down to 2 hours by running all 8 in parallel using DNASTAR Cloud Assemblies. So that's all I brought today. I guess we'll just see if there are any questions at all. All right. I'll just wait a minute here to see if anybody has any questions. So if you're at all interested in some more information about the validation control workflow, we did just have a paper at ASHG. I'm sorry, not a paper. We had a poster at ASHG, and that is available on the website. So I'd be happy to send that to anybody, or I can let you know. So I got a question here about software being available for clinical use. So DNASTAR software is for research use only. It is not approved by the FDA. So thanks for that question, but at this time DNASTAR software is for research use only. All right, I guess with that, I will just feel free to reach out to me. My email is there, and I'd be happy to answer any questions after today. Thank you.